This presentation reviews pH, logarithms, definitions, and pKa. With the goal of functional understanding, some terms are used imprecisely. In clinical practice, we frequently hear the terms proton, hydrogen ion, and hydronium ion. Patients decrease gastric acidity with proton pump inhibitors. To understand these terms, start with a hydrogen molecule containing one proton and one electron. Strip away the electron to obtain a single, positively charged proton. Also called a hydrogen ion, this proton combines with water to become the hydronium ion H3O. The hydronium ion concentration determines pH. pH equals the negative logarithm of the hydrogen ion concentration. This can be rewritten to 1 divided by the hydrogen ion concentration. Because the hydrogen ion is in the denominator, as the hydrogen ion concentration rises, the pH falls. At a low pH, there is a high concentration of hydrogen ions. As the pH rises, hydrogen ion concentrations fall. Logarithms are exponents in reverse. If I asked you, what's 10 to the third power? You would think 10 times 10 times 10 and tell me 10 to the third power equals 1,000. Conversely, the logarithm of 1,000 is asking 10 to which exponent equals 1,000. So the logarithm of 1,000 equals 3. 10 to the second power equals 100. Therefore, the log of 100 equals 2. Finally, if 10 to the first power equals 10, the log of 10 equals 1. It is important to note that logarithmic values indicate exponential change. Because of its exponential nature, small changes in pH represent huge changes in hydrogen ion concentration. A normal patient's pH is 7.40 with a hydrogen ion concentration of 40 nanomoles per liter. A drop of merely 0.3 pH units doubles the hydrogen ion concentration. Dropping to a pH of 6.8 quadruples the acid load. Pay close attention to the curved line in this picture. Bedside tools such as the rules of thumb found in Barish and the base deficit found in other texts approximate using a straight line. These tools lose accuracy at extremes of pH. Click to either explore the issue in more depth or to continue. These lab values come from an M&M meeting some years back. pH 6.83, PaCO2 126, bicarbonate 20.3, base excess negative 9.3. For rational treatment, the clinician needed to estimate both the respiratory and metabolic contributions to acidosis. Frequently used bedside estimates include rules of thumb and the base excess. Barish's clinical anesthesia uses rules of thumb to quickly estimate the respiratory contribution. The metabolic component is calculated based on this result. For every 10 millimeter rise in CO2, the pH drops by 0.05 units. For every 10 decrease in CO2, the pH rises by 0.1 units. In our example, the carbon dioxide level was 126. Take the patient's CO2 of 126 and subtract a normal CO2 of 40. Our rule of thumb states that for every 10 units of CO2 rise, the pH drops by 0.05. We must divide 86 by 10, do the multiplication, and predict a pH drop of 0.43 units to 6.97. However, the patient's actual pH was 6.83. We now know that the patient suffered from 0.14 units of metabolic acidosis. The attending physician made this calculation and then made the poor choice of giving bicarbonate. Base excess and rules of thumb are drawn from opposite sides of the same equation. Carbon dioxide is hydrolyzed by water to become carbonic acid. Carbonic acid donates its proton and becomes bicarbonate, we call carbon dioxide a respiratory acid and bicarbonate a metabolic base. The standardized base excess formula uses bicarbonate to calculate the degree of metabolic disturbance. The formula listed can be found in Miller and should not be memorized. Do memorize that base excess is the number of moles of bicarbonate needed per liter of extracellular fluid to normalize pH. The dose of bicarbonate to give is 0.3 times the patient's weight in kilograms times the base excess. 0.3 represents bicarbonate's volume of distribution. Give no more than 1/2 of the calculated dose. Both base excess and rules of thumb graph straight lines. In this picture, the calculated value using rules of thumb is drawn over actual hydrogen ion concentrations at a given pH. As the pH drops, there is an exponential increase in hydrogen ion concentrations. Our patient had a pH of 6.83. At this pH, our formulas dramatically underestimated the respiratory contribution. The patient's problem was acute bronchospasm with minimal metabolic issues. Bicarbonate should never be given to treat respiratory acidosis. In our chemical equation, we see that giving bicarbonate increases carbon dioxide levels. 100 milliequivalents of bicarbonate produces 2.24 liters of carbon dioxide, or about 10 minutes of normal carbon dioxide production. If you cannot increase the patient's minute ventilation, bicarbonate dramatically increases carbon dioxide levels. This is a problem because while carbon dioxide easily diffuses across cell membranes, bicarbonate does not. Bicarbonate is a charged molecule, and cell membranes keep it from crossing. Carbon dioxide, an acid, freely enters cells without a mediating buffer. This worsens intracellular acidosis. If you cannot ventilate the patient, do not give bicarbonate. In this patient, the bronchospasm was breaking as bicarbonate was given. In spite of the clinician's poor treatment choice, the patient did well. Definitions provide mental images for processes. The two most commonly used definitions in anesthesia come from Arrhenius and Bronsted-Lowry. Arrhenius defined an acid as anything that reacts with water to form hydrogen ions. This stated the obvious without providing a mechanism. The Bronsted-Lowry definition provides a mechanism. Acids donate protons, bases accept protons. Most tests ask students to provide the Bronsted-Lowry definition. However, Bronsted-Lowry is not without problems. Overloading patients with chloride results in hyperchloremic acidosis. However, chloride has no proton to donate. Carbon dioxide is a respiratory acid, but has no proton to donate either. In both of these instances, water donates the proton, but does not cause the acidosis per se. It is worth noting that as long as your patient has water in their body, they have a nearly unlimited source of protons. Strong acids fully dissociate at pH ranges compatible with life. Hydrochloric acid always runs to the right and goes to completion. Weak acids only partially dissociate. In this equation, carbonic acid donates protons as the equation runs to the right. However, bicarbonate accepts protons, causing the equation to run leftward. We consider bicarbonate a buffer because as hydrogen ion concentrations rise, bicarbonate pulls them out of solution. The pKa is the pH at which there are equal concentrations of a weak acid and its conjugate base. In this equation, the acid is written as HA. H stands for the hydrogen ion that is attached to the acid. Since hydrogen ions are known as protons, some texts use the term protonated. On the opposite side of the equation sits the acid's conjugate base. The conjugate base can accept the proton, and the equation can run in both directions. We are now ready for the acid-base titration curve. In the middle sits the pKa, where there is a 50-50 mix of acid and conjugate base. Move 1 pH unit in either direction, and the solution becomes almost entirely protonated or unprotonated. This curve explains why local anesthetics work poorly in infected tissue. Local anesthetics are weak bases, and mild tissue acidosis renders them useless. This issue is explored in the presentation on local anesthetics.